Okay, good morning, students. Am I audible?
Good morning, ma'am.
So that means I'm audible. So, good morning. Good morning. So, last time we did somatic hybridization, right? So, in somatic hybridization, we discussed about fusion of protoplast, selection of hybrid cell, and identification of hybrid plants, right? And we missed one thing in this that the first step was basically that how we are going to isolate protoplast. So, first step should be the isolation of the protoplast, and then fusion, and then the identification of the hybrid cells, right? So today's topic is basically a repetition what we studied last time, but the isolation part, the protoplast isolation part was not covered last time. So I need there's another PPT which in this will discuss about again the isolation. Isolation is the new part, but the fusion and hybrid selection and the uses we have discussed already, right? So I thought that let's again go through this one particular protoplast isolation and fusion, right? So we discussed last time also that in those plants which are not sexually compatible, especially if you want the characteristics from the plant or we want to hybrid a plant, where plants they don't belong to the similar family, right? They are not related to each other. So in a natural process, it is not possible because they are not compatible. But if we isolate protoplast and then we fuse protoplast, then we can select hybrid cells, right? Again, then we studied different methods to select hybrid cells, that how we are going to identify the hybrid cells and how we are going to look for that, right? So today's lecture also focuses on the similar line of the thing. So just once again, we'll go through this topic again. So first was the protoplast isolation. So this we missed last time. So we know the protoplasts are the cells where the cell walls are removed. We don't have a cell walls, right? So definitely, naturally, these things are not there. We need to remove those cell walls and we need to isolate the protoplast in order to go through this process, right? So protoplasts are the cells which have had their cell walls removed, usually by digestion with enzymes, right? So and a number of cellulose enzymes they digest the cellulose in plant cell walls, while pectinase enzyme they break down the pectin holding cells together, right? So once this cell wall is removed, the resulting protoplast it is spherical in shape. Usually, the cell wall basically they provide, or a cell membrane they provide rigidity to the cell, right? They provide kind of a support to the cell. So if we remove the cell walls or cell membranes, so definitely they are not in one particular shape, right? So digestion is usually see how do it? There are a number of methods to remove cell walls. We can use enzymes. We can incubate them in the presence of certain solution of a higher concentration than the cell content, so which will lead to the plasmolysis, right? Again, this will make the cell walls easier to digest. Then debris they are being filtered or they are centrifuged out of the suspension, and protoplasts are then centrifuged to form the cell. So on suspension, the protoplasts can be then further. We can culture them on media, which can induce cell division and the differentiation also. So a number of the plants can be regenerated from single experiment. For example, a gram of potato leaf tissue can produce more than million protoplasts. So see how much easily and how much quickly we can multiply the plant from a very small amount of the starting material. So this is why this is useful, right? So protoplasts again we can isolate them from the number of plant tissues. We can use roots also. We can use flowers, roots, or even the anthers or the pollen also, right? So the isolation and culture media used they can again vary with the species and with the tissues from which protoplasts are isolated. So this starting from the day when we started this course about the plant tissue culture, I discussed this thing that tissue culture media can vary with a different plant. It can vary, right? For one plant, we need one kind of a media, and for another plant, we might need to add something, or we might need to reduce some components. So tissue culture media is required to be standardized for each plant, right? So the isolation and culture media, it again vary with the species. and with the tissues from which we are isolating protoplasts. For example, it could be different for the flowers, or maybe they could differ for the pollens also. Then again, why this is important, or why protoplasts are important? They are used in a number of ways in a research or a plant improvement also, and they can be treated in a variety of ways. For example, electroporation, incubation with bacteria, heat, or to induce anti-take up. So these are the number of the methods basically that how we can introduce some genetic material or some other gene, how we can make them take up DNA. So there are a number of methods for that, right? So protoplasts can be then cultured and then plants are regenerated. And in this way, genetically engineered plants can be produced more easily than is possible using intact plant or intact cell. So in the next coming lecture, we are going to discuss how protoplast culture is being used in the genetic engineering. So we'll discuss some examples where the protoplast culture has been used to genetically modify the plant, or in the genetic engineering techniques how we are going to use it. So this culture is very commonly used in the transgenic studies also, or in genetically engineered plants where we are trying to produce new plants, maybe for a disease resistance or maybe for some other quality in the plant that we want in them, right? So plants from, again, this is one drawback in a natural selection, that plants from distant related or unrelated species are unable to reproduce sexually as their genomes or mode of reproduction are incompatible. So, for example, in one variety, I see one quality, and in other distant related variety, I see that that plant is resistant to particular one disease, which the plant one is not resistant, or in one family. So I want that this plant, I need a plant which is resistant from the disease also, but it is retaining its qualities also, or maybe of a high yield, or maybe of a good color of the crop, or maybe good shelf life of the fruit, right? So this is not possible by the natural crossing, because these plants are not compatible. They belong to a different species. So sexually they cannot reproduce, right? So this can be done by the protoplast isolation method. We can isolate the protoplast from both the plant types, and we can fuse them, and then we can select the hybrid lines. So mostly in one or two hybrid lines, there will be qualities which contains the both qualities of a good yield also and a disease resistant also. So that selection we need to go for, right? So these things, these kind of a processes are important, right? So protoplasts from unrelated species can be fused to produce plants which can combine desirable characteristics such as disease resistance, good flavor, or could be the stress tolerance, or a cold tolerance, or a temperature, other any other environmental stress tolerance also, right? So again, this we studied last time also, that fusion is carried by application of the electrical methods, or by the treatment of many chemicals which are used for the fusion of these protoplasts are called fusogens, right? We used last time we discussed this term, that they are known as a fusogens. So fusion is carried out by the application of electric current, or by treatment of chemicals such as lead. So fusion products can be then selected on media which contain antibiotics or herbicides, and then they can be induced to form fruits or roots, and hybrid plants can be tested for desirable characteristics. This we studied last time also, that how we can select whether desired characteristic has been introduced. So either we use the minimal media, right? For example, some media where the parents individually, if they are some characters are being introduced, so they will not grow. But only the fused products or a fused protoplast will be able to grow on those minimal media. So like this, we can go for the selection. So this is again the same, that fusion protoplast can result in a disease resistance or some good products. And this is about the protoplast. Okay, these are the applications. Okay, protoplast fusion may be used to produce interspecific or intergeneric hybrids also. And this is an important tool for gene manipulation, because it breaks down the barriers to genetic exchange which is imposed by conventional breeding systems. Then again, this fusion technique has great potential for genetic analysis and for strain improvement, and it is particularly useful for industrially useful microorganisms also. So this technique is very commonly used. Then Enzymes are used for breaking of the cell wall. So again, we know that the cell wall of the plant is required to be degraded, so we can use certain enzymes such as bacterial cells wall are degraded by lysozymes, fungal cell walls are degraded by novozymes C34, which includes glucanase and chitinase, and streptomyces cell wall are degraded by lysozymes and acromopeptidase. So different kinds of enzymes are being used. And the fusion of the protoplast, this we discussed last time also, that it could be the spontaneous fusion that the protoplast during isolation they often fuse spontaneously without anything or any requirement. This phenomenon is called spontaneous fusion. And during the enzyme treatment, protoplast from adjoining cells is fused to their plasmodesmata to form multinuclear protoplast, and then there's the induced fusion. Fusion of freely isolated protoplasts from different sources with the help of fusogens or the chemicals are it is known as the induced. So it is a spontaneous fusion and the induced fusion. Normally the isolated protoplasts they do not fuse with each other. So there's a reason that why they do not fuse with each other very easily. Because the surface of isolated protoplast it carries a negative charge, right, around the outside of the plasma membrane. Thus, there is a strong tendency in the protoplasts to repel each other due to the same charge. Since they are carrying the similar kind of a charge, so they repel each other. They do not fuse each other very easily. So this type of a fusion need a fusion inducing chemicals which actually reduce the electronegativity of the isolated protoplast, and then it allow them to fuse with each other. Now the isolated fusion they can be fused. This we discussed last time also that mechanical is a mechanical fusion. So in this process, these protoplasts they are brought together to in a contact with each other under the microscope using some micromanipulator or some micropipettes. So we can just put them together, and then there's a chemofusion. So we chemofusion, as the name suggests, chemicals. So we are using certain chemicals for the fusion process. So several chemicals has been used to induce protoplast fusion, such as sodium nitrate, PEG, or a calcium ion. And the chemical fusions they cause the isolated protoplast to adhere to each other, and it leads to the agglutination, which is followed by the fusion of the protoplast. Right? Okay, then we have electrofusion. So this we discussed the last time also that some mild electric stimulation can be used to fuse the protoplast, but for this we required some equipment, right? Electroporator or some these kind of equipment. So here there are some glass capillary microelectrode. They are placed in contact with the protoplast, and a very small or a low amount of electric field is applied, which it gives rise to dielectrophoretic dipole generation within the protoplast suspension, and this makes them to fuse together. So this can be only done where we have these kind of equipment present with us, right? Then mechanism of a protoplast fusion. So they say that the exact mechanism is not known, but there are several explanations which are being put forward by the people. And they are like when protoplasts are brought to very close proximity, this is followed an induction phase thereby which changes the induced electrostatic potential of the membrane, which results in the fusion. And after the fusion, the membrane stabilizes and the surface potential returns back to their normal state or the formal state. And there are some literature which shows that protoplasts, when the protoplasts are closely adhered, the external fusion causes disturbance in the intramembranous proteins and glycoproteins. And this increases the membrane fluidity and create a region where lipid molecule intermix, which allow the covalence of adjacent membranes. The negative charge which is carried by a protoplast is due to the intramembranous phosphate groups which are being present there. So when we add calcium ions, they causes reduction in the zeta potential of the plasma membrane, and under this situation, the protoplast they are fused, right? So this is again some explanations which are being given, and they say that in fact we use polyethylene glycol. It acts as a molecular bridges connecting the protoplast. So calcium ions link the negatively charged PEG and membrane surface, and on the elution of the PEG, the surface potential are disturbed, which leads to intramembrane contact and subsequent fusion. So these are some of the possible explanations that why the fusion takes place and how the fusion takes place under certain circumstances. Now, this is the diagrammatic representation which shows major steps which are involved in protoplast isolation culture and regeneration of the plant. So, we starting from here, the young plant. So, from a young plant, we are going to take some explants. Explant can be a leaf also or some other parts also. So, we'll the normal tissue culture procedure will surface sterilize them, will peel off the epidermis, will plasmolize the cell in order to yeah, isolate the protoplast or remove the cell walls basically, right? Then we treat them with some enzymes, right? And then protoplasts, they are being isolated. Then isolated protoplasts, they are being plated, and then a cell wall regeneration takes place, and then a callus species form, and again, which will lead to the plant, a whole plant regeneration is being taken up, right? So if there's a question like that, discussed in detail how the protoplast is isolated culture and how plants are regenerated, so this figure is sufficient to explain the steps, right? Just that you might need to add what kind of enzymes or what kind of a method we are using for isolation or the fusion also. So those additions would explain the whole process, right? So a detailed 5 to 8 mark question means a flow diagram or this cycle chart and plus some explanation. So that's all you are required to do, right? And then the applications of a protoplast fusion, this we discussed last time also that protoplasts, they contain all the intracellular organelles of the cells, and it is an important link in transfer of micromolecules between cytoorganelles. Currently, most of the labs which are engaged in a fungal genetics or they are using gene manipulation procedures, they are based on the protoplast. So they are whatever genetically modified plants or a fungal genetics or any other plants also, the protoplast, this is the simplest method which is being preferred by the people. Therefore, to further improve genetic properties of these strains using protoplast fusions are attempts to develop methods for preparation and regeneration of the protoplast. Again, the process involves protoplast metagenesis, transformation, and protoplast fusion. The direct bioconversion of cellulosic material to ethanol by intergeneric fusions between some species of Saccharomyces, they appear to be they are the best techniques for an alternative approaches for the ethanol production. So for biofuel production or for these kind of a techniques also this kind of a technique is being used. Also, this is helpful in production of complete set of cellulases by the protoplast fusion of Aspergillus niger. One produce more amount of endo and exogluconase and other produce more beta glucosinolases. So some other kind of the processes also this kind of a technique is very commonly used. Then these are some of the work done by the people. For example, Gary et al. In 2002, he studied the effect of protoplast fusion on endotoxins which are produced in Bacillus thuringiensis species and found that Bacillus thuringiensis fusions have 1.48 times more endotoxins than the wild type. And according to US patent, fusions of Penicillium crassulaceum and Cephalosporin, they are produced a novel lactam antibiotic. So they are more beneficial. So these are some of the work which are being done by the people that how this technique can be useful for the industrial production of the antibiotics or other enzymes, right? So this can be used. Then protoplasts, they have a wide range of applications. For example, definitely, we can regenerate whole plants from the protoplast. We can go for the production of the hybrid plants from the protoplast fusion type, and hybrid plants can have a desirable characteristics of both the parents. So it depends upon us. We need to choose the parents wisely, and then a hybrid plant where the characteristics of both the parents are incorporated. And it is easy to perform single cell cloning with the protoplast. Then genetic transformation can be achieved through genetic engineering of the protoplast DNA. Then again, these are excellent material for ultra-sector studies also. And isolation of cell organelles and chromosomes is also easy from the protoplast. Again, they are also useful in some of the membranes, very transport and uptake of the processes by the membranes. Then regeneration of the protoplast. That how do we regenerate the protoplast? So protoplast regeneration, which may also be regarded as protoplast development, occurs in two stages. First, the cell wall will be formed, and then the development of the callus or the whole plant, right? See, first we isolate the protoplast, we remove the cell wall, we remove the plasma membrane, and then the protoplasts are isolated. Then the fusion will take place, and then we need to regenerate them. The regeneration will be again require the formation of the cell walls, and then development of the callus, and from the callus the whole plant will be developed, right? The formation of the cell wall. The process of cell wall formation in cultured protoplast starts within the few hours after the isolation. That may take 2 to several days under suitable conditions, right? So as the cell wall development occurs, the protoplast lose their characteristic spherical shape. जैसे वो cell wall remove करने के बाद they become in certain spherical shape. So after the cell wall is formed, so that spherical shape is lost. The freshly formed cell wall is composed of a loosely bound microfibrils which get organized to form a typical cell wall. Now, this process of cell wall development requires continuous supply of nutrients also and maybe some carbon source such as sucrose is the commonly used carbon source which is being used in the different nutrient media. So this will require lot of carbon source also. Then development of callus into the whole plant or the whole plant. So as the cell wall formation around the protoplast is complete, the cell will increase in size and then it will divide. Divisions will start again within 2 to 7 days. And after subsequent division, it will result in a small colonies. And by the end of the third week, visible colonies, which are called macroscopic colonies, they are being formed. Now, these colonies are then transferred to osmotic-free or a sorbitol-free medium for the further development to form the callus, right? Then the callus will be formed. And again, then callus. Once we have a callus, so we know how we can develop a plant from the callus. The subsequent, we again might need media maybe for some composition of some hormones for the shoot formation or the composition of the other hormones for the root formation. With those media, a whole plant can be regenerated, right? So with induction and appropriate manipulation, callus can undergo organogenic or embryogenic differentiation to finally form a whole plant, right? Now these are the some of the example of the plant species which are regenerated from the protoplast. For example, vegetables, cucumbers, sativas, capsicum annum, woody trees also are being generated, ornamentals, rosa species, chrysanthemum, pelargonium, tubers also, beta vulgaris, oil crops, and legumes. So this protoplast are being used to develop or regenerate whole plants by using this process of a protoplast isolation and a protoplast fusion. And plants are being regenerated by this method, right? So as in the beginning it was discussed that a small amount of a potato leaves can lead to the generation of millions of the plants also, because we can isolate the protoplast and from the protoplast we can regenerate the whole plant. So this is again a good method for a micropropagation also, that if we need a number of the plants to be multiplied, from a small starting material, we can go for this. Another important factor is if we want to certain kind of a desirable characteristics to be introduced into the plant. So protoplast fusion, isolation of the protoplast and the protoplast fusion is one of the best methods to go for this kind of a process. And genetic engineering techniques also for the in the plants, this method of a protoplast isolation and a fusion is being very commonly used, right? So with this the process of a somatic hybridization, which involves protoplast isolation, protoplast fusion, selection of the hybrid cells, and the identification of the hybrid plant, it covers everything. So we are done with the somatic hybridization. We are step-by-step procedure basically. So in the last time we missed the isolation of the protoplast. So that was being basically included in today's lecture. So when we talked about the somatic hybridization whole process, so it will involve not 3 aspects but the 4 aspects, which will be the isolation of the protoplast, then will be the fusion of the protoplast, and then will be the selection of hybrid cells, and then the identification of the hybrid plants. So selection of the hybrid cells and identification of the hybrid plants we studied already in detail last time, that how do we select them, how do we select the hybrid plants, how do we know whether the hybrid plants are being formed, and in which we have the desired properties, right? The culture mediums which are supplemented with certain antibiotics, right? Or the minimal culture medium with some introduction of which includes where we include some kind of the chemicals, right? So by those methods or cultured on a selection medium. So these methods can be used to identify the hybrid plants, and there was some mechanical visual identification of the hybrid plants also. That how we can identify hybrid plants by maybe just morphology of the hybrid plants, or there are certain other genetic techniques also by which molecular techniques by which we can we can understand the genetic constitution of these kind of a plants, such as we can go for RFLP using markers, we can go for a PCR technology sequencing and isolation, right? So this we studied last time only, right? So with this we are done with the somatic hybridization also, and this part is covered. And if there's any question or anything, you can please ask your question. And after that, we'll go for the next topic maybe next time, next class, not today's class, right? So if you have any questions or any queries for this especially, please ask your question.
Ma'am, could you please explain the mechanism again? Actually, I couldn't hear at that time.
Pardon?
Ma'am, could you please explain the mechanism again?
Which mechanism? The fusion mechanism or the
Yes, ma'am.
isolation mechanism?
The fusion mechanism.
Okay, fusion mechanism. Okay, so fusion mechanism, I discussed the last time also that for the fusion of the protoplast, we need certain methods. There are certain chemicals which are being used, or they are which are called fusogens, right? For example, polyethylene glycols or the sodium nitrite is a common chemical which is being used, right? Or there is a process called electric field, right? Sometimes we can use the electric field. First is a mechanical fusion. So in a mechanical fusion, the protoplasts they are just brought close together, right? And some using some there are some especially some small micropipettes come. So using those micropipettes, we'll put them together, and by some process, they'll just come together, and the fusion will take place. This is one process. The second process is the chemical fusion. So in chemical fusion, we use number of the chemicals. There are PEG is the commonly used, and sodium nitrate was the other one, and third one was the calcium ions that we use mostly, right? So that can be done. Third one was the electric fusion. So in electric fusion, some very small amount of electric current is just passed through them to fuse the protoplast. But this is being done by using some, we need equipment to be used. Electroporators, those equipments are called electroporation. Electroporation is again a method जब हम competent cell बनाते हैं ना जब cloning करते हैं right उस वक्त भी electroporation we usually use this kind of a method. So again, this is another method where we just generate some mild electric stimulation to fuse these kind of a protoplast, right? So these are the method basically for the fusion of the protoplast. Okay.
Yes, ma'am. Thank you, ma'am.
Okay. So basically fusion हमने last time भी किया था. The one step the isolation was left last time. So for that I repeated this whole that before we go for the fusion we need to isolate also protoplast, because isolation of a protoplast is also a tricky process. We cannot just isolate them very easily, right? Because there are cell walls around and we cannot disrupt them mechanically also, because that will again may injure the protoplast also. So maybe some enzymes we need to use. How we are going to isolate the protoplast. So that was the main aspect of today's lecture. That by carrying out the digestions can be carried out by the enzymes. The different enzymes are used to digest the cell wall, so that can be used for the isolation of the protoplast. Right? Okay, so this is all for today's lecture. And if you have any questions, please go for the questions. Otherwise, I'll just give you one small assignment question, right? I have to give you one home assignment also. I'm still looking for the topic. Okay, okay. So tomorrow you have your plant pathology exam, right?
Yes, ma'am. We had a presentation.
Presentation also an exam also, right?
Yeah, yes, ma'am. Presentation as
So
well as
how
exam.
we'll do it? Okay, we'll do the exam first and then we'll do the presentation. Okay. 1 hour exam. We have two classes, so 1 hour exam and 1 hour for presentation, or maybe one and a half an hour for presentation is sufficient, and rest time we can go for the exam.
Yes, ma'am. That would be fine.
Um, okay. We just two questions, small cases in the assigned. Should be visible. Okay, it is visible now. Please hurry up. Just in your own words, not nothing copy pasted. Just whatever you have understood from this lecture. Okay, in the meanwhile, we can take the attendance also, and I'll take next class, plant pathology just after this class only, right? So we'll meet for plant pathology at 11:20 around, right? So this in this way, the last class जो lunch के बाद है वो save हो जाएगी आपकी, right? And one more thing, that assignment, I can see tomorrow is the 3rd Feb, right? So tomorrow we have two groups. There is a group 2 by Adhish Mishra, and then there is a group by Tanvi and Vivek, right? So, but tomorrow, since we'll have exams, so we'll be able to take only one group. So, which group is prepared for tomorrow? Tell me. वैसे तो आइडली दोनों ग्रुप का प्रिपेयर होना चाहिए. तो शुड आई सिलेक्ट कौन प्रेजेंट करेगा या आप खुद बताओगे? यस विवेक, कुछ कह रहे थे आप?
हम कर लेंगे.
अधीश, ठीक है. सो अधीश वाला ग्रुप कल प्रेजेंट करेगा, एंड आपका जो विवेक वेरा वाला ग्रुप है, लेट मी फिक्स द तन्वी एंड विवेक वाला ग्रुप, दे विल प्रेजेंट ऑन, वेट, 10th, दे विल प्रेजेंट 10th फेब में भी दो है, 17th फेब को एक है. सो विवेक वाला ग्रुप प्रेजेंट करेगा 17th फेब. प्लीज नोट डाउन द डेट, राइट? मैंने अपने से नोट कर लिया है, आप लोग भी नोट डाउन कर लो, क्योंकि 10th को ऑलरेडी दो ग्रुप्स हैं. So they'll present on 17th, because 17th we have only one group, right? Fine, okay. Okay, so we can just take the attendance, and we'll close the meeting, and we'll meet in a plant pathology class. Okay, let's do one thing. We'll take attendance together in a plant pathology, right? I'll download the files from here. So I'll see you at 11:15. We have a class, right? So let's meet at 11:20 around for the next class. Okay.
11:20.
It's 11:50.
No, 12:20. Sorry, 12:20.
Yeah.
12:20 we'll meet, right?
Yes, ma'am.
Okay, fine. I'll see you then at 12:20.
Okay, ma'am.